Thanks for coming. Isn't nature amazing? That was incredible. I'm going to be talking about one species, or maybe two, but it's a different kind of system. So translocation or reintroductions are a commonly used conservation tool. We do this to augment population numbers. We do it for genetic diversification of populations, to assist with plants' delicate life stages, to assist in migration. We do it for mitigation for loss of habitat, for ecological restoration, among other things. And I'm going to be talking to you about so we know that translocations have genetic ramifications, but I'm going to be talking to you in particular about how genetics can inform translocation efforts. And the species that I'm going to be talking about is Hazardia orcutti. It's a perennial evergreen shrub in the Asteraceae, and it lives in the coastal sage scrub. It flowers in the late summer and early fall and has resinous entire leaves. It was first described in 1885 by Charles Orcutt in Baja California, and it was only known from Baja California until 1979, when it was discovered by Oberbauer in San Diego County. So let's look at its distribution. So historically, there were 17 localities reported in Baja California, where recent searches have shown that that's now reduced to 9. And in fact, only 2 of those populations really have substantial numbers. At Punta Banda, you have 750 plants or so, and north of Ensenada, a population of about 300, and the rest are 50 plants or fewer. So then there's the single population in Encinitas in San Diego County. So altogether, that makes fewer than 2,000 natural individuals globally. This does qualify as a rare plant. So it's listed with CNPS as a 1B1. It's threatened on the California Endangered Species list. It was listed in Mexico on their endangered species list in 2010. And it was a candidate for federal listing from 2004 until it was removed from candidacy in 2013. So how's the population doing in Encinitas today? So I show you here the landscape context for the only US population, and which is shown here. It resides in a natural area. The Manchester conservation area is here. It's about 125 acres, and here's a landfill, here's a power line, and here's a lot of development around it. It is adjacent to the San Elijo Ecological Reserve, which goes off the map here. But the area of occupancy for the native population is only 1.5 acres, and with about 670 individuals there. So there's a compelling need to do for some conservation action for this species, I think. And so the Center for Natural Lands Management proposed establishing 4 new populations, which they undertook at the start of the new millennium. So seed were collected from the native population in 2001, and plants were grown in a nursery for 1 to 2 years, and then somewhere between 125 or 200 individuals were planted out at each outplant site. So the outplant sites are here, a bit north. One of the outplant sites here is within the Manchester Reserve, but at the northern end of the reserve, about half a mile away from the native population. One is in San Elijo Lagoon. I note that San Diego Botanic Garden here was not one of the test outplant sites. However, it did receive some of the plants from the same seed collection, which they planted there in 2005. And so those plants are going to come up later in the story, which is why it's on the map, but they're not part of the formal CNLM outplant test. So how are these outplant populations doing at this point? So this is showing the population data from the 4 outplant sites. The dark blue are the adults, the light blue are the recruits. The white are my inferred numbers of adults, since we didn't have population data from every year, and so, but these are adults, so they're probably present. So what you can see is that Manchester, that these two top outplant populations are recruiting, and they're growing populations. Here, La Costa Green had a slight decrease in numbers of adults and then has started to recruit. And at San Elijo Lagoon, a more steep decline, higher mortality when the outplants were put out, but very recently showing some recruitment. So we undertook a genetic study to evaluate the genetic results of the outplanting tests. And we wanted to know whether outplanted populations captured the full genetic diversity of the native population, and whether the population structure differed from that of a single population, which it effectively is when you gather seeds from the wild and plant them out. So we were interested in what happens here. So we genotyped the data using fragment analysis, using ISSRs. We sampled 30 individuals from each population. And we only sampled the original outplants. We did not sample any leaves from the offspring. We used 2 primers and derived 471 loci to look at in the analysis. So in terms of what we found overall that there was low but significant genetic differentiation between populations. And so the idea that this is one homogeneous population is in fact not what we observed. So we found that there was differentiation between the native population and the outplant populations. If we look at this in more detail or in a different way, we also saw that the outplant populations had greater within population variation than the native population. So we found this if we looked at the percent polymorphic loci, or if we looked at Shannon's information index, or if we looked at the number of private alleles. All of the outplant populations showed greater within population variation than the native population. This was a surprising result initially. We also looked at pairwise comparisons and showed that there was significant genetic differentiation between the native population and the 4 outplanted groups. And also between the different outplant populations, which is in a way less interesting, but in particular there was significant genetic differentiation between the native population and the 4 outplanted groups. We also did a Bayesian cluster analysis, and this defines genetic clusters with distinct allele frequencies. And so what you have here is a visual representation of the genetic structure of the data segregated by population. And so here we found that there were 4 genetic clusters that were identified, and those are represented by the 4 colors that you see. And the proportion of the individual that was assigned to a cluster is represented by the height of the bar and the relative proportion shown on the y-axis, and each vertical bar represents an individual. And usually this tool is used to infer ancestry, but because of the nature of this study, we're using it more to see the manifestations of the genetic shifts that arose in the process of outplanting, perhaps, or somehow. And in a completely homogeneous mix, where the outplants mirror the genetic frequencies of the native population, you'd expect the frequency of the genetic clusters to be about the same in all 5 populations. But that's not what we found. Instead, we have, in particular, these 2 outplant populations that have a high proportion of a cluster, which is uncommon in the native population. It's present but uncommon. And we see that that increases in its abundance in those 2 populations. And we see, to a lesser extent, changes in abundance of the frequencies in the other 2 outplant populations. So what could be causing this? Well, there's 2 kind of classes of causes. So one could be the sampling for the population genetic study relative to the collection for the seed propagation. It could just be a kind of a mismatch between what we sampled, which was we know was spatially spread out throughout the population, and we don't know how the seed sampling seed collection sampling protocol went. Secondly, it could be genetic changes from the propagation and the outplanting process. So there could be natural variance in the seed viability between the maternal sources, or there could be non-random seed collection. So there could have been seed collection from a limited number of individuals. Or perhaps there was selection during germination, during the seedling growth in the nursery, something more differential mortality in the greenhouse or in the growing environment. And then when plants are put out for outplant, perhaps there was non-random allocation of plants to the outplanting sites. Or subsequent mortality, once they were planted out, also could have then shifted the genetic frequencies, basically, which we're seeing. There was an additional piece of this story, and that is that Leslie Randall at San Diego Botanic Garden had wanted to do some seed germination trials. So she had gathered a bunch of seed from a Hazardia orcutti mother and had grown it up in pots. And she noted that a bunch of the seedlings looked weird. And in particular, they didn't really look like Hazardia orcutti. So there was the possibility was raised that there could be some hybridization going on. So Hazardia squarosa variety Grandelioides has an overlapping distribution with that of orcutti. And we included 5 seedlings and Hazardia squarosa in our ISSR analysis as just, and it was a very small sample, but it was, since we were doing it anyway, we threw them in. And here's what we found. What you're looking at here is peak profiles from the ISSR fragment analysis. And so what you can see is that here's Hazardia orcutti, here are the seedlings, and then squarosa is on the bottom. And for these two peaks, you can see that they're present in orcutti and in seedling 2 and in seedling 3, but they're missing from seedling 1 and 5. And then likewise for this peak, it's present in squarosa, seedling 5, seedling 1, and also for the other primer, we have similar phenomenon with seedling 4. And so this was pretty clear evidence that their hybridization could occur. At San Diego Botanic Garden, these squarosa was about 30 meters away from the orcutti. So it was pretty clear that this is what had happened. So this raised some concerns about what was happening in the outplant populations. We could have saved ourselves a little bit of lab trouble by looking at the leaves. And so what you can see here is that the leaf morphology does reflect the peak profiles that we saw. I mean, we needed to do that because it could be that those plants with entire leaves were also harboring squarosa genes, but just not in the morphology. But it is a good management tool that maybe you can just look for seedlings that have dentate margins. So what's the future for Hazardia orcutti? Well, there's some good news here. First of all, overall we found that the native population is not showing signs that it's genetically depauperate. So the population number there seems adequate for that. The native population is in a managed protected area at this point in time. And the outplants, which number more than 800 at this point, do provide a valuable buffer from the loss of the natural population here. But threats remain for this species in the United States as well in Baja California, which I'm not really addressing. So there could be catastrophic loss of this very small single population due to fire. There can be gradual loss from human recreation impacts. It's surrounded by housing development, and dogs and bikes are noted as threats to this species. There can be gradual loss from drought, from climate change. But more, probably of greater concern is the possibility of introgression with Hazardia squarosa. And there's confirmed presence of squarosa at 3 of the 4 outplant sites, at one of them within 3 meters of the orcutti, and at 2 of the others at a slightly greater distance. So what does this mean for management? Well, the highest priority, I think, is to minimize the chances of population loss through introgression with Hazardia squarosa and to plant out of pollen flow distance. Do we know what that is? No, we don't. So graduate students find that out. We should aim for maximal genetic representation of source population during seed sampling and keep standardized lineage records during propagation to make sure that things get mixed up and randomized in putting out for outplanting. And more generally, for translocations of rare plants, ideally that would be informed by genetics before they're planted out. I want to thank my colleagues and funding sources, and also give a shout out to Charles Orcutti, who is also making a cameo in the next two talks. I want to thank
You can take one question.
So you mentioned that there's unusually high genetic diversity in two of the outplants, or presumably. Is there any possibility that when plants were collected, they were collected in Mexico as well and put into the outplanting?
No. Oh, the question is, with a higher diversity in the outplants, is there any possibility that any of the seeds were collected in Mexico? And if so, no. I mean,
No.
I've talked to CNLM, and that was not the understanding. So that would have been a very strange thing to have happen. No,
Okay.
these are from the native population in the Manchester Reserve.
Okay.
Yeah.